Acid-fast stain, Ziehl Neelsens methods. Acid-fast stain. Acid-fast stain or acid-fast staining is an example for differential staining. So we told that differential staining means after staining, if you're observing under microscope, we can differentiate organism into two. In acid-fast staining, we can differentiate acid-fast organism and non-acid-fast organism. In that acid-fast staining, Ziehl Neelsens method is the widely popular method used to detect or demonstrate acid-fast on non-acid-fast organism. And the Ziehl Neelsens method was invented or introduced by two German scientists called Franz Ziehl and Adolf Neelsen. So this, by the Ziehl Neelsens method, we can identify acid-fast organism, mainly Mycobacterium tuberculosis and Nocardia species. Acid-fast staining, Ziehl Neelsens method procedure. In this Ziehl Neelsens method, first we will add the primary stain, that is strong carbol fission, to the smear, and after that we will heat the smear until the vapors are arising. So what will happen during this heating of this smear along with this stain, that is strong carbol fission, the bacterial cell wall is generally thick in the Mycobacterium tuberculosis and other acid-fast bacilli. So while heating, the cells of this organism, the bacterial cell wall of this organism will become thin so that the stain will easily enters into the bacterial cell wall. And after that, we will decolorize the stain with sulfuric acid or acid alcohol. And depending upon the organism, this percentage of the sulfuric acid will be different. Here you can see in this Mycobacterium tuberculosis, we use 20% of sulfuric acid. In Mycobacterium leprae, we use 5% of sulfuric acid. And Nocardia species, we use 1% of sulfuric acid. And acid-fast parasite like Cryptosporidium, Cyclospora, and Microsporidia species, we use 1% sulfuric acid. And in bacterial spore, we use 0.25 to 0.5% of sulfuric acid. That means depending upon the organisms, the percentage of sulfuric acid or the decolorizer will be vary. And the last step is the counterstain with the methylene blue. And we will add this counterstain methylene blue. And after that, we will observe the organism under oil immersion microscope. So the first step is the primary stain. We use strong carbol fusion. And after adding this strong carbol fusion, we will heat the slide until the vapors are arising from the slide. So by seeing these vapors, we can identify that the smear or is already heated well. And we will add decolorizer, that is sulfuric acid. Depending upon the organism, it will vary. And after the decolorization step is over, we will add counterstain with the methylene blue. And the peculiarity of this acid-fast organism is this acid-fast organism will resist decolorization with this acid alcohol or sulfuric acid. So the acid-fast organism will be seen in red color, that color of carbol fusion, and the other acid-fast, non-acid-fast organism will appear in blue color. That will take the color of this methylene blue. So this is the acid-fast staining of Mycobacterium tuberculosis. Here we use 20% of sulfuric acid as decolorization agent. So this is the pink or red colored rod-shaped bacteria in blue background. This is the sputum smear. So the red color bacilli can be seen in blue background. That is the red color is the color of strong carbol fusion, and this blue color is the color of methylene blue. And this is the acid-fast staining of Nocardia species. The Nocardia species are long or branched filamentous-like organism. So in Nocardia species, we told that we used 1% sulfuric acid as decolorizer. So here you can see this is the red-colored filamentous bacteria in blue background. So this is the acid-fast staining of Mycobacterium leprae, and you can see this red-colored bacilli, that is intracellular bacilli in red color in blue background we can see that is the red color is the color of strong carbol fission and blue color is the color of methylene blue. In Mycobacterium leprae we use 5% sulfuric acid as decolorizing agent. Acid fast stain principle. Mycobacteria and Nocardia do not stain with simple stain, so these organisms can be stained by heating them with carbol fission. The heating helps the stain to penetrate to the cells. Once the organism has taken up the carbol fission, they are resist decolorization by acid alcohol, that is sulfuric acid. That is why it is called as acid fast bacteria. And this acid fastness is due to the high lipid content, that is mycolic acid, in the cell wall of this organism. So the main principle of acid fast organism is the presence of mycolic acid in the bacterial cell wall, and this is called acid fast bacilli because it will resist decolorization with the acid alcohol or sulfuric acid. Once it has taken the color of primary staining, that is strong carbol fission, and we told that the carbol fission we will heat until the vapors arising from the slide. So during heating, the carbol fission will penetrate into the cell. It will trap into the mycolic acid. The mycolic acid is present. It will go and bind to the mycolic acid. Once it is stained with this strong carbol fusion, that is primary staining, it will resist decolorization with the acid alcohol, that is sulfuric acid. That is why it is called as acid fast bacilli, and the main principle behind this acid fastness is due to the presence of mycolic acid in the bacterial cell wall.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.